other and vice versa. So it's basically almost as if the cells shared a single mind, that they can communicate with one another by just sharing the same kind of content. Things can pass through these gap junctions. You'll see this a lot in actually muscle cells too. And so this molecule can diffuse over here, other molecules can diffuse over here, and these gap junctions are made by proteins called connexins. You don't really need to know that word, but you need to know that these connexins are not always open. They can actually be regulated to close and open. And so these cells can decide when to open, when to close, when to let things pass, when to close off the barrier. So there's also decisions around here too that can be made. Also, gap junctions are the only way to have an electrical signal propagate to one cell to another directly. There's no other way in physiology currently to directly have one cell's electrical current pass from one to another without inducing some other change in the other cell. And this is because they're sharing the same cytoplasm, they're sharing the same water essentially. And so the electrical impulse or signal can travel through this water, through the gap junctions, and affect both cells. Now, next one we have is contact signaling. This is pretty much used in developmental systems or immune systems, and the idea is that ligands are on the surface of a cell, and it comes in contact with another surface of a cell, and they come together, ligand and protein, just like this, and this creates a signal. And so immune cells will do this all the time. They will, glob onto something, and they will detect for its glycoproteins, and if it doesn't match up, it's going to do phagocytosis beneath the cell. If it does match up, it can recognize the cell as being part of the host, it will ignore it. So that happens a lot of time. And also in development, when cells are sorting around as development occurs, this is also very important to figure out who your neighbors are and what they're doing. Diffusion still works here, though. Why?
Because they're close to each other.
Let's say these cells are stationary. Diffusion's still happening. How is it still happening? Remember, cell membrane is under the fluid mosaic model. What about the fluid mosaic model? What does it say? No, not quite. It says, not quite. It's the idea that proteins in the membrane can move around the membrane in a fluid manner. And it's shown that it's true for most proteins, not all proteins. Some proteins are anchored, but they can move around. They're fluid. That's why it's called a fluid mosaic. It's mosaic because you have a whole arrangement, a whole array of proteins, but it's fluid because they're all moving around. And so this ligand and this receptor are diffusing. They're moving around through random movement, through entropy, but it's along the surface of the membrane. It's diffusing around the surface. So instead of diffusing in a volume, it's diffusing around a plane. That's the difference. All right, the last type of short distance signaling is autocrine/paracrine signaling. And so this is the idea that one cell kind of spits out a signaling molecule, and it travels in the extracellular fluid and interstitial fluid, and it can go find the receptors of close by cells. And so the signal, when it affects other cells, is called a paracrine signaling. So this is paracrine for others. And autocrine, when it affects itself. And you may ask, what's the point of putting out a molecule if you're just going to be signaling yourself? And it turns out it's actually a pretty important process for, once again, for immune systems and development to kind of confirm your identity. If you put out a signal that says, I'm an alpha cell, and then you keep on receiving that same signal, it reaffirms the cell's identity, so to speak. And so there are special cases where autocrine is really important here. And so an example of short distance signaling is the signaling molecule histamine, right? Whenever, if you ever take a needle and just scratch yourself, that part swells up. It swells up into something called a heel, actually. Sorry, a wheel. How do you? It's a wheel. That's kind of like the red raised portion that comes off from whenever you scrape yourself, or with a needle or something like that. That is basically paracrine signaling. The breaking of those cells release histamines. Histamines then will go affect neighboring cells, so it will affect capillary beds and other cells to then basically, for the capillaries, will open up, it'll make those capillaries more permeable, that fluid rush into the area. That's how you get swelling in those damaged areas. And then it will inform immune cells to also rush into. And so that's all a local response, and that's all done by paracrine signaling. Alright, then we have long distance signaling. So this is when basically the signal has to go a distance that's much farther than the diffusion can do practically. And so diffusion can only really happen like on the on the millimeter, maybe even centimeter scale, but if you want to go any further than that, you have to do some long distance signaling. And one main way of doing it is endocrine signaling, and we'll talk a lot about endocrine systems in the next lecture Thursday, so just keep that in mind. The whole idea of endocrine signaling is you put your signals, you put your lichen, you put it out like you would in paracrine signaling, but instead of just diffusing in the ECF, it gets picked up by the bloodstream, it travels in the bloodstream, and then it also diffuses out of the bloodstream and affects cells. And here's the thing, one signal, once it's put out in the bloodstream, essentially all cells will be exposed to it, right? It's kind of like someone sent a blast email. Everyone's going to see the email. The same thing here. When those endocrine signals enter the bloodstream, all cells are, as long as they have a connection to the bloodstream, will be exposed to the signal. Now, not every cell reacts to that signal. And the reason that is because some cells have a receptor for that signal, while others don't. And so that's how you mediate that. That's how you mediate how some cells will react to a certain signal while other cells don't. It's either based by the presence or absence of that receptor. And so a signal that meet up with that receptor will, make that cell do some kind of response in return. There are a couple other ways of doing long distance signaling which are similar to endocrine signaling, but not exactly the same. One is through the nervous system signaling. So this is basically, and we'll talk about this in a couple of weeks, next week really, where a neuron will get an electrical signal that goes down its axon, and as its axon terminals will then release neurotransmitters that will diffuse and target and reach a target cell to create a response. This is considered long distance signaling because these neurons can travel very long distances. They can travel upwards to a meter. So you can have really long neurons. You can also chain a lot of neurons together to go far distances as well. And so this is basically the idea of just taking electrical signals, passing it long distances, and then converting it to what will become eventually a chemical signal, which will then affect a cell and create a response. You have a slightly different variant of the nervous system, and the neuron here is, instead of giving out a chemical signal to a cell, it will dump that chemical signal into the bloodstream, and then it will do something very similar to any other endocrine signal. It will then travel the bloodstream and then affect certain cells. And so this is a special type of endocrine signaling. We call specifically these signals neurohormones, because they're working a lot like hormones that would be produced by a gland, like the pancreas or the pituitary gland, but instead of being created by a gland, it's being created by a neuron. That's why we call them neurohormones.
Aren't they still considered ligands at this point?
Every signaling molecule is a ligand. Mm-hmm.
But what is the neuron releasing? Is that a neurotransmitter?
Yeah, the punchline is they're releasing some kind of chemical. And the fact whether they're called neurotransmitter versus neurohormone is basically what's the target. If it's going into the bloodstream, it's considered a neurohormone. If it's going to go to some cell and a synapse, then it's a neurotransmitter. So yeah, it's more or less the same thing, but they're classified as different things based off where they're going. Yes?
So then are they classified under the nervous system signaling still? Because it's using a neuron?
Yeah,
But then
it is.
technically it's still like using endo.
It's acting like an endocrine, yeah. It's still part of the nervous system because it requires a neuron.
Right.
And as we'll talk about later towards the end of this lecture, there is a lot of gray areas between the endocrine system and the nervous system. There's a lot of cooperation between the two systems to create signals. And so it's going to get fuzzy, but because technically it uses a neuron, it's part of the nervous system.
So it just kind of depends on what's sending out the signal.
Yeah.
Okay.
No, you can have.
Not necessarily.
No, you're right with that statement. But you can, I'm getting ahead of myself, but basically you can have coupled systems where a neurohormone then triggers endocrine cell, that then puts out a hormone, and so then you have this nervous/endocrine pathway. And we'll talk about that towards the end. But yes, you can have those mixes, but this is technically considered part of the nervous system. Alright, and so that signal, whatever it is, whether it's a closed signal like a paracrine signal or it's a hormone traveling through the bloodstream, this is the pathway they follow. The signal molecule will try to bind to some kind of receptor. Sometimes it's a membrane, sometimes it's not a membrane. And then it will activate some kind of intracellular response. So something inside the cell, that's what intracellular means, will happen because the receptor is bounded to its ligand. It's going to create some change. And that change is going to affect some protein, which will in turn create some